Hey everyone, welcome back to the channel. Now, if you've been following my recent videos, you'll probably know that I've been diving deep into the world of bioprinting. In our last video, I explored the first two processes of the processing stage, which were inkjet bioprinting and extrusion-based bioprinting. Well, today we're going to take it to the next level, and I'll be showing you the two other advanced bioprinting techniques, which are laser-assisted bioprinting and stereolithography. Now, we'll go through how each of these methods works, and then I'll move on to the post-processing stage. First is laser-assisted bioprinting. Now, this technology uses lasers in order to precisely deposit tiny droplets of bioink, which allow us to build complex biological structures. Now, let's go through this step by step in order to see how this exactly works. First is the laser and the mirror. Now, the laser generates a focused beam, and then the mirror reflects it in order to direct it to a specific target area. Next, the beam passes through a focusing lens. Now, this part is really critical because the lens concentrates the laser's energy onto a tiny area on the donor side below. That focused energy is what gives us the power in order to create tiny controlled droplets of the bioink layer. Now, next is the donor side itself. Now, this slide holds a layer of bioink, which is basically a mix of biological materials and living cells, as we have discussed before. And on top of that, it has a thin metal layer that absorbs the laser's energy. Now, here's where things get really important. When the laser hits that laser-absorbing metal, it creates heat. This heat vaporizes a small bit of the bioink underneath, which creates high-pressure bubbles. And what does this bubble do? Well, it pushes a tiny droplet of bioink down and out of the donor slide. Now, let's talk about this printed droplet. Now, as each laser pulse creates one droplet, so we get this super precise control. Now, the droplet lands on the collector slide, which is where the structure gradually builds up layer by layer, and we can use this in order to replicate anything from simple cell patterns in order to complex tissues. Now, let's look at the advantages and disadvantages of laser-assisted bioprinting. First advantage is that it has high precision. We can print individual cells with micrometer accuracy. And therefore, it's very ideal for essentially printing complex structures. And also, it is nozzle-free. Therefore, it avoids clogging and reduces cell stress, which I think is very important because we're not dealing with any type of ink, we're dealing with bioink. Also, it's a non-contact method, therefore it has minimal cell damage and there's zero contamination risk because there's zero physical contact. Also, we can handle bioinks of large ranges of viscosities, and therefore we can also handle bioinks with perhaps dense cell suspensions. However, some of the disadvantages of laser-assisted bioprinting include the fact that it is very expensive and also complex. It has high setup costs and it requires precise calibration. Also, the choices for bioink is essentially very limited. This is because the bioink must be laser absorbent. And it's also very slower for larger jobs because it deposits one droplet at a time. And also, it does have thermal risks, given the fact that the laser heat can affect the cell viability. Okay, now next is stereolithography, also known as SLA. Now, first we have the laser source and the scanner system. The laser source generates a focused beam, and this is often a UV or a visible spectrum light. And the scanner system directs this laser beam to a specific point on the bioink surface, and we can pretty much assume that the scanner system is basically acting as the mirror in the laser-assisted bioprinting technique. Now, the bioink responds by polymerizing. or solidifying precisely where the laser hits, creating a defined layer of our structure. And what do we call this type of ink that kind of solidifies under laser exposure? We call this photopolymerizable bioink. Now let's move on to the platform, or the Z moving component. Now the platform starts really close to the surface of the bioink and it moves incrementally along the Z axis. And in each layer cycle, after the laser cures a layer, the platform moves up or down depending on the setup, allowing fresh bioink in order to kind of flow over the cured layer and create space for the next. Now the laser systematically polymerizes each layer based on a pre-programmed 3D model. And as each new layer bonds to the last, the final printed object gradually forms, allowing for high-resolution construction of complicated 3D molecular structures with smooth surfaces. Now, let's kind of move on to the disadvantages and advantages of SLA. First advantage, it offers exceptional resolution and smooth surface finishes, which kind of makes it ideal for applications where fine structural detail are crucial, such as perhaps tissue scaffolding, or even organ modeling. However, on the downside, SL requires photopolymerizable bioinks, which kind of limits the number of materials that you can use because not all bioinks solidify under laser exposure. It's a very relatively slow procedure for larger structures due to the sequential curing of each layer, which can make it less practical for having to bioprint large volumes. Okay, now we've explored all 4 of the processes of the bioprinting in the processing stage. Now let's kind of move on to our final stage, which is the post-processing stage. In the post-processing stage, the tissue undergoes cross-linking. Now this involves chemical or physical cross-linking where bonds are formed within the bioink in order to increase the sort of structural connection. UV light, temperature changes, or chemical agents can be used depending on the bioink's properties in order to do the process of cross-linking. After that, the tissue is left to mature. This is a very time-dependent process and it's performed under tightly controlled conditions and typically in bioreactors, which are essentially devices or systems that provide suitable and regulated environments for cell growth and production. So it's very regulated and it provides suitable conditions for cell growth. This is to mimic the in vivo environment, given that bioprinting is an in vitro process. Now, for those of you who may not know what in vivo and in vitro means, in vivo means inside a living organism, in vitro means in a lab dish or a test tube, essentially not in a living organism. Okay, and that brings us to the end of our 3-episode bioprinting lecture. Hope you enjoyed it and see you next time.